Now, the other issue in terms of, so the issue of whether your risk of PML returns to zero if you're off the drug for a period of time or not, it's just not known. We just don't understand that. So at this point, I would say that it's not rational to give patients drug holidays to try to reduce the risk of PML, because we have no biological evidence that that makes sense, plus we have this risk of the rebound phenomenon. The other question is what do you go to when you go off Tysabri? Right now, that's one of those $64,000 questions. We personally tend to go to Gilenya. The reason for that is that we initially were trying to use Copaxone or the interferons. We tend to use Copaxone more because it's a little bit easier for us to use on our side. It's fewer side effects. We don't have to worry about liver problems and depression and other things. But in fact, in the country, many physicians tend to use interferons the same way. So it's just a matter of style. But what we found, though, is when we went to those drugs, we hoped they would maintain the Tysabri level of treatment effect. So Tysabri sort of be an induction therapy, and then you can maintain it with a safer therapy. That's still a valid study design, but there's a paper published about a month ago that said, unfortunately, it doesn't work, that the disease will reactivate within about 6 months, regardless of whether you're on interferon or Copaxone or not after Tysabri. So that's why we tend to go to Gilenya. So far, we've not had anybody who went to Gilenya that's had the rebound effect, because we tend to start it within the first few weeks after you stop the Tysabri. But I do have colleagues that argue that you should go through a washout period from the Tysabri before you start the Gilenya. The problem is they're not including this risk of the rebound effect. Plus, we know the MS reactivates within 3 to 6 months. So I don't see a reason to put a person at risk. And pharmacologically, there's no particular reason why the drug should interact. The issue is that you're switching from one immunosuppressive therapy for the brain to another immunosuppressive therapy. And the question is, will that increase your risk of opportunistic infections? It's possible, but I think the probability is quite low. And so I think we need to observe patients carefully and monitor things very closely. But those are the choices. And that's why it's a very complex decision.
You have a lot of patients that have now done the Tysabri to Gilenya.
So the question is, do we have a lot of patients? We have about 125 that have gone from Tysabri to Gilenya. But it's very early. And we're learning by the seat of our pants, unfortunately. Again, because I think the company could do more to help us learn more rapidly of what's the best strategy here. But it takes time. There's all sorts of issues why that's been hard to do. There's a question over there?
Yes, rather than a standalone therapy, would a drug combination have a more effective result?
So she's a plant. I actually paid her $5. So the question is, rather than using a therapy as a monotherapy, what about combination therapy? In my view, that's the future of MS. So we have 9 partially effective drugs with very variable safety profiles. By end of next year, we'll probably have 12, maybe 13. And they'll have the same problems. I think I know pretty much every drug that's at least hit phase 2. I think I know most of the drugs that have hit phase 1. There's no drug currently in development that I know of that has biological properties that suggest it's curative. And from the standpoint that you guys think about it, where you get treated for a certain period of time, then you stop and the disease is gone, and you don't get it again. So we're going to be stuck with this class of drugs of long-term therapy. So if the disease on average starts around age 30 and then it burns out by age 65, that's 35 years that we have to treat people. And the probability is that the longer we use these, particularly these more potent drugs, like teriflunomide, alemtuzumab, rituximab, fingolimod, that we are going to see increasing safety problems. You can't shut down the immune system and not eventually run into trouble. The remarkable thing is we can shut down parts of it and maybe take 5 or 10 years before we run into major problems. So there are opportunities there. And I think that the opportunities are basically what you're suggesting. And the question is, can you use a staged approach where you use an induction therapy to try to get a high-level treatment effect in a way that using that therapy is quite safe? For example, with rituximab. Rituximab used over a 2-year period of time is remarkably benign. And so that's a drug we can use for 2 years. Then the question is, follow it up with something. Now, because I mentioned that with Copaxone, because it tends to produce B cells that are protective, one possibility would use a drug that kills the B cells that tend to drive the disease, rituximab, and then use a drug that generates a new population of B cells that might increase your resistance to disease reactivating, which is Copaxone. And so we actually have that study funded, actually funded through Teva. It's an investigator-initiated study that I designed. It's a single center that we'll do here. We'll study 200 patients, and just to see whether that works. So the rituximab will shut the disease off for the first year or so, So the question we have to ask, will the disease stay off of patients that are on Copaxone for the next year, for year 2 and year 3? And that's what we're going to try to test. There also are many other combinations. So BG12, because the way that it works is being combined with Avonex and with Copaxone. Laquinimod there's now discussions going on to do it in combination trials. Daclizumab is already a combination trial, again with Avonex. Probably also makes sense with Copaxone. And then these drugs here, Laquinimod and BG12, in my view, are becoming very attractive as the maintenance therapy because their safety profile is good, they're a pill, they're easy to take. And then that opens up all of these other ones as being the induction therapies. And the other question is, we don't know exactly how these work yet. It may be that they're complementary in the way they work and we could add them together, or maybe we could add one of these to Copaxone or Rebif or Avonex or something and get a major effect that way. So yes, those are the major approaches that I think we'll be pursuing next year. They're not going to be driven by companies though, by and large, unless they own both products. So it's going to have to be driven probably by investigator-initiated studies mostly.
In the HIV marketplace, do they do the studies in combination before drug companies started paying for them, or were they starting to pay for them before these combination therapies were done? Because now the combination therapy is really the standard of care.
Yeah. So the question is in the HIV world, the AIDS world, how did they end up with combination therapies? It's actually a very interesting story that actually has relevance to you. And AIDS and cancer are the 2 areas where combination therapy has been remarkably effective. So most cancer therapies are combination therapies, and AIDS is certainly a combination therapy. The history of that is that back when AIDS began to emerge in late 80s, yeah. It was about 85 or 86 when the first trial started, about in the early 90s, and they began to accelerate. And at that time, they were being developed like every other drug. So every company had their drug, the retroviral drug, and they were using it as a monotherapy in the studies. And it was the patients and families that came out and advocated and said, we want to use the kitchen sink. We want you to give us everything that's possibly available. And then if it works, we'll then go back and sort out which one's doing the job. We don't want to have a drug that maybe slows the disease by 5 or 10%, which is what most of them were showing. We want to actually get a cure. So we want combination therapies. And it was that pressure that initially led to an NIH study, and then the standard was that when the first drug was identified as having some efficacy, that everybody would be on that, and every other drug would have to be added onto that. That's actually not an unusual paradigm. That's the way we develop new drugs for seizures. We always add the new drug onto the pre-existing drugs and see whether the combination does better than the old drug does. That's what we need to do in MS. We need to identify which one of these therapies should be our baseline drug, and then see whether we can add other things to it to make it better. And it's still early. I mean, the data is just evolving, but that is a key discussion that has to take place. And again, I think that physicians are so concerned, particularly neurologists, so concerned about being out front that they're going to wait for somebody else to lead. And the issue, like I mentioned with Tysabri, if you have a negative JCV antibody test, my colleagues are not going to jump out there and get on national news and saying we think Tysabri should be a first-line drug for people who are antibody negative. They're going to wait for the FDA to act on the company's application. And, but you can. You can affect physicians, and that's why we have these kinds of programs, is because it's important in the American healthcare system, and probably worldwide, for you to be empowered with information that you can then discuss with your physicians, and in that way, the physicians begin to recognize what your goals are, what you would like to see done, and what kind of risk you're willing to take, and that begins to modify their behavior as well.
I know you're also doing research on estrogen and Copaxone.
Yeah.
And that's going in a different direction.
Yeah. So the question is about looking at estrogen-type compounds combined with Copaxone. And it's actually a trial of estriol. You can buy estriol in the grocery store. It's an old estrogen compound. And it was based on work done by a colleague of mine out at UCLA who was looking at it from the standpoint that we know that women are almost twice as likely to get MS as men are. And during pregnancy, the inflammatory disease in the brain and the relapse rate goes down. And then after delivery, there's a rebound effect. So it's always been discussed that maybe estrogen-like compounds or progesterone-like compounds are having some immunological effect. And I can tell you a lot of us have spent a fair amount of our time in the laboratory trying to prove that, and we just can't do it in the animal model. But there was done in a human trial, estriol as a monotherapy, and it did seem to have a modest effect. And the biology of it seemed to suggest that it might be a neuroprotective effect. And so there is an ongoing NIH-funded study of adding estriol onto Copaxone to see whether it improves maintaining brain volumes and decreases loss of function, so prevents disability. So that is an ongoing trial. It is open. It's only open to women for obvious reasons. And you have to have started Copaxone within the previous couple of months or so. But it is, the safety profile is pretty well known, and it is a reasonable trial.